So I asked you for the 2 purines. Let's see what you came up with. Adenine and guanine, right? Remember AG, pure silver kills werewolves, right? Purines. First one was no, A and T, that's the base pairing rule. That's a purine and a pyrimidine. Remember the 2 pyrimidines are C and T, King Tut or Cleopatra Tut or Cats and Tut, pyramids. But A and G, right? A and G are the 2 purines. Purines are double carbon rings. Not Hershey and Chase. I don't even know what that is. I didn't say Puritans. I said purines. So a nucleotide is made up of 3 parts. Nucleotides are the monomers of nucleic acids. We understand that they have a 5-carbon sugar called deoxyribose, a phosphate group, and a nitrogen-containing base. The phosphate groups in the sugar molecules of the nucleotides are going to link together to form the backbone of the DNA strand. What type of bond holds the 2 sides of DNA together? It is covalent. See the side here? These are all covalent bonds. What are the types of bonds here in the middle? Hydrogen. Are hydrogen bonds strong or weak? Weak. The way I remember this is I think about water. Yeah, exactly. You dive through water, right? You separate water. No big deal. Those are hydrogen bonds. You go dive into a tree or a wood floor, it doesn't work so well, does it? All those, yeah, ask that person. I won't repeat the name of the child who was not wearing a seatbelt. When he hit covalent bonds, it was a whole different story than if that was water in front of him, right? Yeah, right? Okay. That poor kid, I give him a hard time every day about not wearing a seatbelt. I almost made him cry yesterday. I got to stop. Yeah. So I know, I got to quit picking on him. I keep telling him every day, I'm like, did you wear your seatbelt today? And he goes, yes. So the 5-carbon sugar in DNA is called deoxyribose. Everybody say deoxyribose. That's a sugar, right? Okay, so DNA's name, its long name is deoxyribonucleic acid. So DNA is named after its sugar. Everybody say ribose. And now say ribonucleic acid. That's RNA, and RNA is named after its sugar. Everybody say adenose. And that's adenosine triphosphate. That's ATP. That's another nucleotide, right? So those are all named after their sugars. So if you look at DNA, and we talked about this a little bit already, we understand that DNA is made of these monomers up here, which are called nucleotides. Every nucleotide will have a phosphate group, a 5-carbon sugar, and if they are DNA, the carbon sugar will be deoxyribose. It will have a nitrogen base, which in DNA will either be G, C, T, or A. The 2 purines, AG, the 2 pyrimidines, CT, pure silver, pyramids, King Tut or Cleopatra Tut, however you want to remember it, I don't care. But you also have to know that you always have a purine with a pyrimidine, or a pyrimidine with a purine. So that means that if C is present, G must be there. T is present, A must be there. So you always find T and A and G and C. Any questions on this? Purines are double ring structures. Pyrimidines are single ring structures. The 2 purines are adenine and guanine. The 2 pyrimidines are thymine and cytosine. A goes with T, G goes with C. I always remember at G and C, just to keep it straight. Must you remember all of that? Yes. Everybody say, that is the base pairing rule. A, T, G, C. We copy DNA onto? RNA wiggles through goes out into the, to find a, to make a, by assembling, and forming. What type of RNA wiggles through a nuclear pore? Everybody say messenger RNA. mRNA. Okay. So the information in DNA is contained in the order of the bases. Well, the base pairing structure allows the information to be copied. How do we copy? G to C and T to A, or A to T. Anytime you have a G, C, C, G, A, T, T, A, T, A, G, C, C, G, C, G, A, T, T, A. Doesn't matter what it is. You always know what the other one is. Okay? So in DNA, each nucleotide has the same sugar group and the same phosphate group, but each nucleotide might have a different nitrogenous base. I could say nitrogen, I could say nitrogenous. I'm referring to GCT or A, purines and pyrimidines when I say that. The four kinds of bases are called adenine, guanine, thymine, and cytosine, and you understand that A and G are purines and have double ring structures. How do I remember this? Ag is the symbol for silver. Silver is pure. It kills werewolves. Ag. Yep, don't say AU. That won't make sense. No, I know. It will later. When I get to RNA, we'll get to that. There is a U that comes in RNA. But then the bases T and C have a single ring structure, and these are our pyrimidines we talked about. T and C. Pyramid reminds me of Egyptian, the King Tut, or Cleopatra is also there for the C you guys came up with, I think. So a purine on one strand of the DNA molecule is always paired with a pyrimidine on the other strand. You always find a purine with a pyrimidine, a G with a C, a T with an A. Specifically, adenine pairs with thymine and guanine with cytosine. The base pairing rules are dictated by the chemical structure of the bases, meaning you only get A with T and G with C. The hydrogen bonds between the bases keep the two strands of DNA together. Now, knowing what I told you about water, if I pick water up, what happens to it? It falls apart because those are weak hydrogen bonds. Would DNA stay together if it weren't wound together in a helix? No, the two strands would what? Separate. Okay? The weak hydrogen bonds, you got it. Exactly, you're making my point for me. So looking at a strand of DNA, on the side you will have sugars and phosphates covalently bonded together. This is the strong part of the ladder. Think about a ladder. It's not going to break where you hold on to the sides. Where's it going to break? Where you stand, if it's going to break at all. And that's this weaker area in the middle, the hydrogen bonds. You will notice that you have 3 hydrogen bonds between G and C and only 2 between T and A. That is why T cannot bond with a different purine. T will never bond with G. No, because of the bonding capabilities of each. So we need to know that DNA is made of monomers called nucleotides, and each nucleotide contains a phosphate group, a sugar, and a nitrogen base. The nitrogen base can change. It can be a purine or a pyrimidine. A purine is an A or a G. A pyrimidine is a T or a C. Hopefully I'll have repeated this so many times that you'll never forget it. Yeah, they won't bond. They think of them as 2 puzzle pieces that only bond together in a certain way, and if it's not the right puzzle piece, no dice. So paired bases are said to be complementary because they fit together like puzzle pieces, and because of the base pairing rules, if the sequence of bases is known for one strand of DNA, we automatically know the base for the complementary strand, and it can be quickly identified. Let me show you what I mean. What if I told you that I have 10% adenine in my DNA? I want you to think about that for a minute. I'm not going to ask you on Peardeck, but I'm going to ask you one at a time. I'm going to see if you can figure this out. I want to know how much percentage of guanine, thymine, and cytosine I have. I told you how much I have for adenine. So if I have 10% for adenine, how much percentage of my DNA is going to be thymine? Yes. Also 10%. Anybody else agree with that, or you guys disagree? Okay. So if that's the case, and if my pen will work right, okay, how much guanine do I have? 40 and 40, I heard it. So what you're saying is if Mr. Mason gives you just one of the bases percentage-wise, you know the rest of the story, correct? There's a guy named Chargaff. Everybody say Chargaff. Chargaff figured this out. Okay, he figured out if I had 10% adenine, I also had 10% thymine. If I had 40% guanine, I'd also have 40% cytosine. And then he figured out, since there's only 4 bases there, if I know one, it's very easy, since we have that relationship, to figure out the rest. And that made identifying nitrogenous percentages and how much of each is there possible. Now, that doesn't seem important initially, but it's very important if you're trying to learn and figure out the base pairing rule, because he didn't know it. We'll talk more about how it came about here in just a second. So Watson and Crick, these are the two most famous DNA science people because they're attributed with figuring out the actual shape of DNA. But they used work from Chargaff and then these other two scientists called Wilkins and Franklin. It was Rosalind Franklin that actually did most of the work. She didn't win the Nobel Prize, though. Wilkins did because he was a guy. She's now attributed for the work, though. Well, it's not funny. It's pretty, we're not Iran, We're more civilized than that. What? Yeah, I don't like Iran. Yeah. What? Oh, I ran into the store the other day. I was hungry.
What are you talking about?
I don't know now. They went off on a sidetrack with me. So, well, no, that's an expression. Like I ran up to the store real quick. And Iran's a country. They're bad, evil.
Oh, Iran.
We went from the Middle East to like Family Fare or Kroger real quick. All right, so Chargaff shows that the amount of adenine always equals the amount of thymine. We just talked about that. Okay, and the amount of guanine always equals the amount of cytosine. If I tell you one of the base pairs, you'll know the rest immediately. Now, Franklin and Wilkins developed X-ray diffraction, and this means that they looked at DNA underneath a microscope and then took pictures with an X-ray. We'll talk about what they got here. But it suggested that the DNA molecule was actually a helical shape. So these guys determined the shape of it, but they didn't know it was two strands. And then Chargaff, they figured out there were equal amounts of G's with C's. You put those two together, you get a double helix, right? We'll show you what I mean. What if I told you that they used to think DNA was a triple helix? They used to think it was three strands. And that's why they couldn't figure it out. There was actually work published, like textbooks that were published that labeled DNA as a triple-stranded molecule. Do you understand now why they had such a hard time trying to figure it out? Because it'd be like you being taught that DNA is double-strand and it really being single-strand. You wouldn't be able to accept it. So for a long time, scientists didn't want to change their way of thinking. But then Rosalind, she took X-ray pictures of DNA underneath a microscope, and what she kept getting that she couldn't make sense of was this. Anybody know what I'm looking at there? X's and O's. Anybody know what that is? What? Yeah. It's the top view of DNA. She was actually looking at the top view of the double helix, and she was seeing this and the top. Okay? So, seems kind of weird, but when you think about what she was actually looking at, she was looking at the helical structure just coming through a two-dimensional X-ray image. And she didn't have the aha moment. It was actually Watson and Crick that figured out what she was looking at. We'll talk about that right now. But she did, along with Rosalind, Wilkins, and Franklin, they did determine that it was a helical shape. Now, Watson and Crick used both Chargaff's data and the X-ray diffraction studies to create a complete three-dimensional model of DNA. So Watson and Crick it was James Watson and Francis Crick. They're sitting in their office with Post-it notes all over with G's, C's, T's, and A's. And then they had the aha moment when they looked at Chargaff's rules and applied it to Rosalind and Wilkins' X-ray diffraction. And their model showed a spiral staircase in which two strands of nucleotides twisted around a central axis. And that's what we built the other day. And that's what we found DNA to actually be now. It's kind of funny. They didn't do any of the work themselves, but they had like oh, geez, look, it's this, right? Quick summary. I'll let you guys work on your review and keys. DNA is the primary material that causes inheritable characteristics in related groups of organisms. I want you guys to think about this for a minute. Put your pencil down for just a moment. Look up at the ceiling. Okay? Put your pencil down. I'll give you time. Look up at the ceiling and imagine 100 base pairs that go from the ceiling to the floor. G's, C's, T's, or A's. Doesn't matter what. Okay? It's a lot, right? We probably had 20 yesterday we played around with, maybe. Now, imagine it's 1,000. Now imagine it's 10,000. Now imagine it's 100,000. Now go to 1,000,000. Now you're like in space now, right? Okay. Now go to like 100 million. Go to 1 billion, which is 100. Now just think about 2.2 billion base pairs. That's what you are. Every cell of your body has 2.2 base pairs in a row in a little tiny strand of DNA. So now think about how small DNA is, but how big it is, right? So now, if I handed you 2.2 billion different letters, and each one, you had 4 different types of letters, right? Sorry, but 100, 2.2 billion of them. How many combinations would you make? If I gave you all your own set, would any of you make the same? No. How about let me just hand you 4. Let's make it simple. What if I gave you 4 different letters? How many combinations of those 4 could you make? Everybody say 64. 64. Now just add 1 more, right? Now you're looking at a totally different number that gets astronomical pretty quick, doesn't it? So 2.2 billion base pairs in every single nucleus, in every single cell of your trillions of cells inside of you has that complete set of DNA, except for red blood cells. Okay? And if you have 2.2 pairs, you actually have 4.4 billion nitrogenous bases, right, because they're pairs. So now, that's just humans. Some animals have 100,000 base pairs. Some have 1 billion. Some have 5 billion base pairs. So do you understand why life is so different now? I mean, think about it. The possibilities are endless. Now, we have the same types of nucleotide sequencing, which means we have the same proteins. We don't have the exact same versions of those proteins, though, do we? Right? So now imagine this. You got this big strand of DNA that's divided into 46 different chromosomes. And then you look at one gene at a time. Let's say I need some brown eye color. So I copy the gene from DNA onto RNA. What type of RNA? Everybody say mRNA. Everybody say mRNA. mRNA wiggles through goes out into to make a protein by assembling amino acids and forming, and then it makes a protein that gives me brown eyes. So this happens inside of you over 30,000 times. You have 30,000 different types of proteins that make you, okay? And it takes 2.2 billion base pairs of information to code for those 30,000 different proteins. We are complicated individuals, aren't we? Think about it. Ugh. That's just one organism on Earth, right? There's other things. So 3 major experiments led to the conclusion that DNA is the genetic material in cells. Griffith, what did Griffith do? Everybody say transformation. What did Avery do? He proved that DNA was the transformative molecule, right? Say, Avery proved Griffith's experiments. And just say DNA so you remember. Hershey and Chase, everybody say bacteriophage. Say viruses. So they dealt with the viruses. They found out it was the DNA that was the active component in viruses, not the protein, right? And then the DNA molecule is shaped like a double staircase and is composed of 2 parallel strands of linked subunits called nucleotides that form a double helix. The information in DNA is contained in the order of bases, that's in ours, while the base pairing structure allows the information to be copied. We copy it by matching G's with, everybody say C's, T's with, let's do this together. You guys say it after me. T, not that. T goes with A. A. Everybody say T. G. C. Everybody say C. C. G. A. T. C. 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 G. T. A. 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 T. G. C. C. Oh. C. G. A. T. T. A. All right, that's just fun. So Watson and Crick used information from experiments by Chargaff, equal amounts of A's with T's, G's with C's. Wilkins and Franklin, the X-ray diffraction, and they determined that DNA was a double helical structure, 2 strands wound around a central axis. Everybody say Watson and Crick. They are the most famous DNA scientists, okay? All right, you guys.